Welcome to the Deep Dive. We're here to unpack complex sources, cut through the jargon, and, well, synthesize the key knowledge you need. Today, we're diving into just the sheer variety of ways viruses replicate. It's really a fascinating look at microscopic ingenuity, maybe even subversion. And we're starting with something maybe a bit surprising from the sources. Feline leukemia virus, FLV. I mean, think about it. Cats and humans, we live side by side very closely. And this virus, FLV, it uses a phosphate transporter that's really common on our own cells, superconserved receptors. So you'd think, right, with that easy entry point, why aren't humans getting FLV all the time?
Yeah, it's a perfect example to kick things off. FLV can actually get into a human cell. That receptor is there, it works. But, viral success, it's way more than just opening the door. The courses talk about this intimate and prolonged molecular conversation needed between the virus and the host. It's not just entry, it's the whole process.
Ah, okay, so the conversation breaks down.
Exactly. Almost immediately after getting inside a human cell. The door's open, sure, but the internal environment, the host machinery, it's just not compatible. Maybe it's transporting the viral genome, maybe it's expressing the right genes at the right time, or assembly or release. One of those steps, maybe more than one, it just hits a dead end.
Yeah.
It really highlights that viruses need this incredible molecular specificity for every single stage of their life cycle.
Right. That really sets the stage. We're not just looking at, the greatest hits of viruses. We're exploring this huge spectrum of strategies, all evolved to get around these host barriers. And to make sense of it all, virologists use, well, mainly two classification systems.
That's right. You've got the official one, the ICTV taxonomy. Think of it as the formal address book. It puts viruses into orders, families, genera, uses those standard endings like nifirals for orders, and it focuses heavily on physical stuff. What's the genome made of? DNA, RNA, single or double-stranded? Is there an envelope? What shape is the capsid? Physical traits.
But then there's the Baltimore system, often more useful day-to-day for understanding how a virus actually works, right? Tell us about David Baltimore's approach.
Yeah, the Baltimore system is all about function, utility. It kind of ignores what the virus looks like and says, okay, let's group these into 7 categories based on one thing only. How does the virus make messenger RNA, viral mRNA? Because at the end of the day, tricking the host ribosome into reading your instructions, your mRNA, that's the key goal for any virus.
And fundamental to that Baltimore system is knowing the difference between positive strand or plus-strand and negative strand genomes. This applies to RNA viruses and also single-stranded DNA viruses.
Absolutely critical distinction. So the positive strand, the plus genome, you can think of it as basically being the mRNA itself. The sequence is the same. It's ready for the ribosome, more or less. The negative strand, the genome, it's the complement, like a template or a photographic negative. It has to be copied into a positive strand first before any proteins can be made.
So that immediately tells you something important.
Right. It tells you if the virus needs to bring its own special enzyme, one the host cell doesn't have, to make that first copy.
Okay, let's dive into the first big group then. The double-stranded DNA viruses, dsDNA. They're often seen as maybe the most straightforward kind of mimicking host processes, but they still have some amazing tricks.
Yeah.
We've got 2 great examples from bacteriophages, viruses that infect E. coli.
Yeah, perfect contrast. Let's start with bacteriophage T4. It's like the brute force specialist. Its entry is pure mechanical engineering on a microscale. It lands with these long tail fibers, the base plate settles onto the cell surface, and then bang! The tail sheath contracts like a syringe, pushing a central tube right through the bacterial cell wall, injecting its DNA inside.
Just punches right through. And once the DNA's in, T4 goes on the attack immediately, right? What's its first move against the host?
It's basically biochemical warfare. Very early on, T4 makes a protein that tells the host's own enzyme, RNase, to start chopping up all the host mRNA. Yeah, it's essentially shouting, stop everything you're doing, all your ribosomes, all your building blocks, your nucleotides, they're mine now. Total resource grab.
And it even protects its own DNA, chemically, from the bacteria's defenses, the restriction enzymes.
It does. This is really clever. T4 swaps out the normal DNA-based cytosine for a modified version called hydroxymethylcytosine, or HMC. And then often it adds a sugar molecule to that HMC, that's glycosylation. This modified glycosylated DNA is like invisible to the host's restriction enzymes, which would normally chew up foreign DNA. It's a molecular shield.
So T4, get in, take over fast, protect yourself. Now, contrast that with bacteriophage Lambda. Lambda is different. It's a temperate phage. It has a choice.
Yeah.
Kill, now the lysogenic cycle, or wait, Integrate, the lysogenic cycle.
Lambda is, yeah, much more calculated. The genetic philosopher king, perhaps.
Yeah.
When its linear DNA gets inside, it circularizes first using these sticky ends called cohesive ends. And that big decision, lytic or lysogenic, it all hinges on a really elegant regulatory circuit. A key player is a protein called C2.
Seed on C.
If conditions are good, CII levels get high. It's protected by another protein, CNI. And high CI basically signals, okay, let's go for lysogeny.
And if CII is high, what does that trigger? Which genes get turned on?
Two crucial ones. The integrase gene and the CI gene. Integrase is the enzyme that literally cuts and pastes the viral DNA into the host chromosome. At that point, we call the integrated virus a prophage. And the CI gene encodes the Lambda repressor. This protein is the guardian of lysogeny. It binds to specific spots on the phage DNA and physically blocks almost all other phage genes from being transcribed. It enforces the dormant state.
So it just sits there quietly.
Yeah.
Until. What makes it switch back? How does it get induced to go D-lytic?
Stress. Usually DNA damage to the host cell, maybe from UV light or certain chemicals. This activates a host repair protein called RECA. Now, activated RECA has an interesting side job. It interacts with the Lambda repressor, CI, and causes CI to basically chop itself in half.
Ah, self-destruction.
Pretty much. So CI levels plummet. When the repressor is gone, other phage genes can finally be expressed. One of the key ones is the cis gene encoding excisionase. Excisionase works with integrase to reverse the integration process. It cuts the prophage back out of the host chromosome, and boom, the lytic cycle kicks off. It's a beautiful molecular switch.
Incredible control. Okay, let's move from bacteria to eukaryotes. Things get more complicated now, dealing with the nucleus, organelles. Let's take herpes simplex virus 1, HSV1, causes cold sores.
Right. HSV1 is a great example because it shows both types of infection clearly. You have the productive intention, that's the active itcholytic phase, making lots of virus, causing the sore. But then it establishes a lifelong latent infection, usually hiding out quietly in nerve cells and neurons. The viral particles aren't detectable, but the genome is there, ready to reactivate later.
And how does it even get its genome into the nucleus of our cells?
It hitches a ride. The incoming nucleocapsid, the protein shell with the DNA inside, gets transported along the host cell's own internal highway system, the microtubules, using motor proteins like dynin. It travels right to a nuclear pore, injects the DNA, and the linear DNA circularizes inside the nucleus.
HSV1 also has this really weird way of getting its envelope, doesn't it? Not the typical budding from the cell surface.
Yeah, it's quite unique, a two-step process. First, the nucleocapsid buds through the inner membrane of the nucleus, picking up a temporary primary envelope. Then, it fuses with the outer nuclear membrane, losing that first envelope. Then the naked capsid travels through the cytoplasm and acquires its final mature envelope by budding into membranes derived from the Golgi apparatus or endosomes. Then it gets out via exocytosis. Very different.
And what about keeping it quiet during latency in those neurons?
The sources mention a couple of things. One is small, non-coding RNAs made by the virus that seem to actively repress the lytic genes. Another key factor is a host protein called HCF1. In most cells, HCF1 helps activate philic genes, but in neurons, it seems to be kept out in the cytoplasm, unable to help turn on the lytic cycle.
Fascinating. Okay, now for the real giants of the viral world. The Nucleocytoplasmic Large DNA Viruses, NCLD viruses, sometimes called megaviruses. How big are we talking?
We're talking. Visible with a regular light microscope big. Genomes that are huge, 500,000 base pairs, even over 1 million, comparable to some small bacteria.
Whoa.
And what's really striking is their autonomy. They are incredibly self-sufficient for viruses. They encode tons of their own replication machinery, DNA polymerases, RNA polymerases, transcription factors. Some even encode things you normally only associate with cells, like tRNA molecules and the enzymes that attach amino acids to them, the aminoacyl tRNA synthesis.
That sounds almost like a cell. Why aren't they just considered, I don't know, super reduced parasitic bacteria or something?
It's a great question, and it pushes the boundaries. But they still lack 3 fundamental things that define cellular life as we know it. 1, no ribosomes. They still rely entirely on the host ribosomes to make their proteins. 2, no way to make their own energy, no ATP synthesis machinery. And 3, they don't reproduce by cell division, like binary fission. They still assemble particles within the host cell, obligate intracellular parasites, but yeah, incredibly complex ones.
Really blurring the lines. Okay, let's pivot now. Single-stranded DNA viruses, ssDNA. What's their main challenge?
The biggest hurdle is that single strand. Host DNA polymerases, the enzymes that copy DNA, they need a double-stranded template to work properly. So the very first thing an ssDNA virus has to do is get its genome converted into a double-stranded replicative form, or RF. It has to rely on host enzymes to make that complementary strand.
And parvoviruses like B19 that causes fifth disease, they're really dependent on the host for this, right? They're tiny.
Extremely dependent. Parvoviruses have hardly any enzymes of their own. They have to infect very specific cells, often red blood cell precursors. And here's the clever bit. They actually force the host cell to pause its cell cycle in the S phase. Why? Because S phase is when the host cell is naturally synthesizing its own DNA.
So all the host's DNA replication machinery is active and available?
Exactly. The virus basically says, hold it right there, and hijacks all those host enzymes and nucleotides that are conveniently laid out. And they have another trick for replication itself. They use these palindromic sequences, repeats at the ends of their genome, that fold back on themselves to form a hairpin loop. This hairpin acts as a primer, a starting point, for the host DNA polymerase. It's called rolling-hairpin replication. Really ingenious way to get started without needing your own priming enzyme.
Okay, rolling hairpin. Got it. Let's move into the world of RNA viruses. Now, for all of these, there's one enzyme that's absolutely essential, right?
Yes. The star player for RNA viruses is RNA-dependent RNA polymerase, or RdRp.
RdRp. And why is it so crucial?
Because host cells simply do not have an enzyme that can make RNA copies from an RNA template. Our cells make RNA from DNA, or DNA from DNA. Making RNA from RNA, that's a virus specialty. So every RNA virus must either bring the RdRp enzyme with it, prepackaged in the virion, or its genome must be able to be immediately translated by host ribosomes to make the RdRp right away.
And this RdRp, it does two jobs.
That's right. It acts as a transcriptase, meaning it makes viral messenger RNA, mRNA, from the RNA genome template. And it acts as a replicase, meaning it copies the entire RNA genome to make more genomes for new virus particles. Multitasking enzyme.
Let's look at double-stranded RNA viruses first, like rotavirus, a major cause of gastroenteritis. They have dsRNA. Why does the RdRp have to be packaged inside?
Because double-stranded RNA is a massive red flag for host immune systems. Cells have sensors specifically to detect dsRNA as a sign of viral infection. So rotavirus keeps its segmented dsRNA genome safely tucked inside a core particle, the double-layered particle DLP, even while the RdRp inside is transcribing it to make mRNA, which then exits the particle.
So it never exposes the dsRNA directly to the cytoplasm.
Exactly. And the actual replication of the dsRNA genome and assembly of new particles happens within these specialized inclusion bodies in the cytoplasm called viroplasms. Kind of like mini viral factories offering protection.
Okay, now for the plus-strand RNA viruses. Poliovirus is a classic example. Their genome is plus RNA, so it can act like mRNA right away.
Correct. The moment it enters the cytoplasm, the host ribosomes can theoretically bind and start translating it.
But wait, eukaryotic mRNA usually needs that special 5-foot cap structure for ribosomes to recognize it. Polio RNA doesn't have one. How does it trick the ribosome?
Another clever workaround. Poliovirus RNA has a highly structured region, usually near the 5-foot end, called an Internal Ribosome Entry Site, or IRES. You can think of the IRES like a structural landing pad. Its complex 3D shape allows the ribosome to bind directly to the RNA internally, bypassing the need for the usual 5-foot cap. It's a cap-independent mechanism.
Sneaky. And once translation starts, polio uses the polyprotein strategy.
Yes. The ribosome translates the entire RNA genome end-to-end, creating one single gigantic polyprotein. This polyprotein then contains viral proteases, enzymes that cut proteins. These proteases then proceed to chop the polyprotein itself up into all the individual functional viral proteins needed, structural proteins, the RdRp, everything.
Why do it that way? Seems complicated.
It ensures that all the proteins are made in the correct relative amounts, kind of stoichiometrically linked, and it all happens within another specialized structure the virus builds, the replication organelle, or RO, which it constructs by hijacking host cell membranes, like from the ER, provides another layer of protection and concentration.
Okay. We see different strategies in other plus-strand viruses, like tobacco mosaic virus, TMV, in plants. How does it make its different proteins?
TMV uses a couple of tricks. One is making subgenomic RNAs. These are shorter mRNA molecules that are transcribed from internal start sites within the main genome, allowing access to genes downstream. The other trick is read-through. Sometimes the ribosome encounters a leaky stop codon. Most of the time it stops, but occasionally it just reads right through it, continuing translation to produce a longer fusion protein.
And how does TMV spread between plant cells with those thick cell walls?
It uses specialized viral movement proteins. These proteins interact with the plasmodesmata, the small channels that connect the cytoplasm of adjacent plant cells, and actually modify them, making them larger, allowing the viral RNA or even particles to pass through.
Clever. Okay, finally, the negative strand RNA viruses. Influenza is the famous example. Their genome is RNA, so it cannot be translated directly.
Absolutely. This means they must package their RdRp enzyme inside the virion. It has to come in with a genome.
And influenza has that segmented genome, right? Several pieces of RNA.
Yes, usually 7 or 8 segments of negative strand RNA. Each segment typically codes for 1 or 2 proteins.
But the really unique thing about influenza's RdRp is how it makes mRNA, this cap-snatching mechanism.
Cap snatching. It's brilliant in a devious way. The viral RdRp goes into the host cell nucleus. It finds host pre-mRNAs, which have that 5-foot cap structure. The RdRp then acts like molecular scissors. It cleaves off the 5-foot cap along with a short stretch of about 10-15 nucleotides from the host mRNA.
It seals the cap.
It literally steals the cap and uses that stolen capped fragment as the primer to start synthesizing its own viral mRNA.
Wow, so it gets a primer and it messes up host mRNA translation at the same time.
Exactly. Double benefit for the virus. It kickstarts its own protein production while simultaneously inhibiting the hosts. Very efficient takeover strategy. Then the finished virions bud out from the plasma membrane, taking a piece of it as their envelope.
Okay, that covers the main RNA virus groups. Our last major category is defined by a truly remarkable enzyme, reverse transcriptase, or RT.
RT is the defining enzyme for 2 groups. The retroviruses, like HIV, which have RNA genomes, and the hepatinoviruses, like Hepatitis B, which actually have DNA genomes but use RT in their cycle.
Let's focus on RT itself first. What makes it so special? It does multiple things.
It's a molecular powerhouse. It has at least 3 different enzymatic activities packed into one protein. First, it's an RNA-dependent DNA polymerase. It reads an RNA template and synthesizes a DNA strand. That's the reverse transcription part. Second, it's also a DNA-dependent DNA polymerase. It synthesizes the second DNA strand, using the first DNA strand as a template, to make double-stranded DNA. And third, it has RNase H activity, which degrades the original RNA template strand once the DNA copy is made.
And critically, it's known for making mistakes, right?
Very much so. RT lacks proofreading ability. It makes errors relatively frequently when copying the genome. This high mutation rate is a key reason why viruses like HIV evolve so rapidly, leading to things like drug resistance.
So for HIV, a retrovirus, RT takes the incoming plus-strand RNA genome,
Converts it into double-stranded DNA in the cytoplasm. This dsDNA then travels to the nucleus and gets integrated, basically pasted, into the host cell's own chromosome. This integrated viral DNA is called a provirus.
and that provirus just stays there, becoming part of the host's genetic material. How does HIV manage to get all its different proteins made from that one integrated piece of DNA?
Through really sophisticated layers of gene regulation. It uses alternative splicing, extensively cutting and pasting the mRNA transcript in different ways to create many different protein coding messages from the same DNA sequence. It also uses things like ribosomal frame shifting, where the ribosome slips slightly during translation, changing the reading frame to produce a different protein, and read-through of stop codons similar to TMV. All these tricks allow a compact genome to produce a complex array of proteins.
Amazing complexity. Now, the other group using RT, the hepatoviruses like Hepatitis B, HBV, they actually have a DNA genome to start with, but it's weird, gapped.
Yes, the HBV genome inside the virion is mostly double-stranded DNA, but it's circular, and one strand has a nick and the other strand is incomplete, leaving a gap. Very unusual structure.
So how does RT fit in if it starts with DNA?
It's a really fascinating cycle. First, when the virus enters the liver cell nucleus, host repair enzymes fix that gapped DNA, sealing the nicks and filling the gap to create a perfect, stable, covalently closed circular DNA molecule, the cccDNA. The cccDNA is the master template. Host RNA polymerase transcribes the cccDNA to make viral mRNAs for proteins, and it makes a full-length plus-strand RNA copy called the pregenome RNA.
So DNA to RNA.
And then reverse transcriptase gets involved. The pregenome RNA moves to the cytoplasm, gets packaged into a developing viral core particle along with RT. Inside that core, RT uses the pregenome RNA as a template to synthesize the new gapped dsDNA genome.
So it goes DNA, RNA, stance DNA using RT. Wow.
Exactly. The RT essentially makes the DNA genome that will go into the next generation of viruses using an RNA intermediate. And that stable cccDNA template persisting in the nucleus is the major reason why completely curing chronic hepatitis B is so difficult. It's a permanent source of new virus production.
What an incredible tour of viral strategies. From the elegant on-off switch of Lambda to the brute force of T4, the RNA tricks like cap snatching, and the sheer power of reverse transcriptase. It's really mind-boggling.
It really is. And if you think back to those giant NCLD viruses, the megaviruses, it drives home that constant interplay between structure and function. Every unique genome type, every challenge posed by the host, requires a specific molecular tool or strategy, whether it's RdRp, RT, and IRS-modified DNA bases. And those NCLD viruses, with their massive genomes, their self-sufficiency, encoding parts of the translation machinery, they really do make you pause and think about the definitions. Does their existence fundamentally challenge how we define a virus? Could they represent something else entirely, maybe an evolutionary bridge, or even deserve consideration as, a potential fourth domain of life alongside bacteria, archaea, and eukaryotes? It's definitely something researchers are actively debating.
A really provocative question to ponder. We hope this deep dive has illuminated the intricate and frankly brilliant world of viral replication for you. Thank you so much for joining us. We'll see you next time.